Neuroscience has long chased a specific, almost impossible dream: the ability to actually watch thoughts fire in real time inside a living, conscious brain. Seeing exactly how active neural circuits process information is the only direct way to understand both healthy cognition and the physical breakdown caused by diseases like Alzheimer's. But a living brain is an optical mess. It's packed with water, lipids, and tangled cellular membranes that scatter incoming light in a million different directions, completely blocking our view. Researchers have actually been able to bypass this. For over a decade, they've used chemical clearing agents to render dead, preserved brain tissue completely transparent. Applying those same harsh chemicals to a living, breathing subject, however, would instantly destroy the tissue and kill the animal. To get a clear window into a working mind, researchers had to find a way to manipulate the complex physics of light without disrupting the fragile biology of life. This diagram illustrates a concept called the refractive index. If you drop glass marbles into water, you can still see them because the water and the glass bend light differently. But drop those same marbles into heavy oil and they seem to vanish. The oil and the glass share the exact same refractive index, allowing light to travel straight through without scattering. The brain operates on the exact same principle. If researchers could replace the fluid surrounding the neurons with a liquid matching the tissue's refractive index, the entire organ would turn clear. The physical hurdle is that fluids with the correct optical properties are highly dense, sugary solutions. This is the reality of osmosis. When these dense solutions surround a living cell, they violently pull the moisture out of the membrane, causing the tissue to dehydrate and die. To avoid this, scientists needed massive, spherical molecules. Large molecules generate much less osmotic pressure, meaning they can alter how the tissue handles light without crushing the delicate cells inside. Achieving transparency requires satisfying strict optical physics while keeping the biological environment perfectly intact. For a year, researcher Shigenori Inagaki systematically tested nearly 100 different spherical polymers. Every single one successfully bent the light, but ultimately failed by disrupting normal cell function. Working late in the lab one night, Inagaki made a desperate guess. He grabbed an expensive, highly pure laboratory reagent that researchers rarely use for this kind of live tissue work. That chemical was bovine serum albumin, or BSA, a highly soluble protein naturally found in blood serum. When applied to living brain tissue, the opaque gray matter turned completely transparent within an hour, and the cells remained perfectly healthy. The very protein that flows through the animal's veins provided the exact molecular structure needed to bypass the optical barrier. The team named their new solution SeeDB-Live. By pairing it with fluorescent calcium indicators, chemical tags that light up every time a nerve fires, they can map brain activity directly. Researchers can now look straight through the outer tissue, observing activity deep in layer 5 of the cerebral cortex in a living animal. Here, fluorescent signals from neurons become 3 times brighter, making deeply buried structures far easier to record. But the most crucial feature of SeeDB-Live is its biological compatibility. Because albumin is natural to the body, bodily fluids simply wash it out of the extracellular space within a few hours. Once the albumin flushes out, the transparent brain gently fogs back up and returns to its natural opaque state without sustaining any permanent damage. This gentle reversal allows scientists to use the exact same subjects repeatedly over 4 months, tracking how specific neural networks shift and adapt over long periods. By finding a way to safely turn living minds transparent, researchers now have the physical tools to watch diseases disrupt neural networks as it happens and observe directly if experimental drugs help heal them.